Good morning, everyone present at C 2020. I'm Juan Morales. I'm going to talk about gene expression of flavanone 3-hydroxylase, anthocyanidin synthase, and p-coumaroyl ester 3-hydroxylase in tzimbalo fruit. The Solanum caripense is a phylogenetically complex plant commonly named tzimbalo, mostly white and widely distributed in Ecuador and Peru, also present in Colombia, Bolivia, Venezuela, Costa Rica, and Panama. This is an herbaceous plant native of the Andean region. Sucrose, vitamin C, and minerals are higher for Solanum caripense sections for many of these compounds compared to modern varieties of Solanum muricatum and other related wild species. These compounds are important for its high nutritional value for human health. Chimpalito and chimbalito are poems from Ecuador that reflect the importance of culture knowledge, as well as ceramic representation of pepino fruits by Nazca and Moche cultures in Peru. This is slide for formation purposes. At the Salesiana Polytechnic University, in vitro propagation of tzimbalo, the research, and then the fruits obtained by the cultivation of the plants for the conservation and utilization of natural resources. The current work aims at contributing with biotechnological tools for plant breeding programs and emphasize expression on Solanum caripense genes. The objectives describe morphological variation on Solanum caripense ecotypes, identify gene expression of F3H and ANS associated to anthocyanins, analyze gene expression of C3H associated to chlorogenic acid. Here you can see the anthocyanin biosynthetic pathway. We focus on F3H gene and ANS gene. This gene is required for the production of purple pigments. Chlorogenic acid biosynthetic pathway, we focus on C3H gene that is present on eggplant and potato, for example. These genes are important because are related to antioxidant activity as well as anthocyanins. Relative quantification assumes an optimal doubling of the complementary DNA. The amplification efficiency is close to 1, and the target gene quantity is normalized to a reference sample, and it is relative to a calibrator sample. Materials and methods. Here you can see the fruits of the ecotypes and the areas of origin in Ecuador and Peru. The list of morphological descriptors used for Solanum caripense ecotypes. Gene expression by retrotranscription followed by semi-quantitative PCR and retrotranscription followed by quantitative PCR. Components and amplification program used for this study. Here is important to notice that this equation is fundamental to determine the relative expression and the mean fold change. Morphological description by conglomerates analysis and principal component analysis. Gene expression, here RNA was isolated from fruit skin and fruit flesh. Results and discussion. The phenogram represents a cophynetic correlation coefficient superior to 0.8. This indicates a good representation of the similarity matrix. Here we can see that descriptors, fruit stripes, fruit flavor, seed diameter, fruit surface by additional color are qualitative descriptors that have higher eigenvector values or relative contribution coefficients, which means that this can be useful for breeding purposes. And here, for quantitative descriptors, you can notice that fruit length is a descriptor with eigenvector values higher than others, as well as fruit blossom length and fruit blossom width. However, these descriptors are better to evaluate by decomposition of genotype, environmental, its interaction effects. Gene expression, RT-PCR. In this image, we can see that the transcripts of F3H in vivo LTG1 were identified by electrophoresis in agarose gel. The other images shows that the transcript were present and quantified using the weight marker with known concentration. Here, the transcript for bio-LTG1 and here the transcript for bio-CY-B1. Here did not return significant difference, the ANOVA, but it could be because of the nature of the chill. It could be overall reactions, RT-qPCR. This is a tool of higher specificity, and here we can see the mean fold change and the log2 transformed values for symmetrical scale. Here, the difference was significant, and we can see that in this image. The mean fold change of C3H expression for day 14 was 6.24, and it was significantly different from that calculated for day 0, 3.32, and similar for day 5, 4.52. Transcript level of C3H expression increased in 2.92 units after 14 days in post-harvest conditions. Conclusions. The morphological description of tzimbalo ecotypes indicates that the mentioned descriptions are characters that contribute more to the variability, and these are agronomical distinctive to be utilized in breeding programs. The expression of the genes of interest in this research by RT-PCR constitute an analysis applied to the exploration of candidate genes for subsequently transcript quantification in real time. The expression levels of C3H was significantly different. This opened the possibility for selection of genotypes that demonstrate good performance in front of different crop conditions. The approach of candidate genes and its expression represent a promising tool for introduction of tzimbalo into plant breeding programs, focused on the conservation and utilization of Andean resources, as shown in these images correspond to the observation of the fruits that expressed anthocyanin pigments related to the gene ANS. Thank you very much. Reference, I appreciate your comments and questions.